So we're going to go ahead and get started. So let me share my screen here. Display. Come on, there it goes. Okay, so this week, fun stuff. You have your exam, your first exam. It's coming up at 4. So you'll have, let me edit this. It's not due Friday. I lied about that. Available Wednesday and due Saturday. So your first exam will become live later today at 4 o'clock to be precise. So if you click on this now, it won't actually be take you to the exam because it's not ready. Like it hasn't just popped up. Now, in terms of where you can find the exam, you just kind of mosey on over here. You go to tests, quizzes, and your exam would also be available here. Again, it's not available quite yet. So you'll have it at 4 o'clock, and then you'll have a full 72 hours to do it. So it's become available today at 4, which means that it'll be due not Thursday, not Friday, but it'll be due Saturday at 4 o'clock. So you have full 72 hours to take the exam. Now, I know the syllabus says you only have 24 hours. I decided to give you more time. I would just, I thought you would appreciate more time. So you'll get essentially triple the amount of time that you were expecting to get, which I think is, it's always a benefit. Now, in terms of the format of the exam, it is 40 multiple choice questions. You're welcome. I try to please whenever I can. I know, and I also know exams are very stressful for some of you, and I recognize that it's not fun. So, and I don't like giving exams, as I told you guys in the first class, but it's part of life for a college student, unfortunately. It is 40 multiple choice questions, and you use something that it's open book, it's open internet. You should work alone. Now, I can't monitor whether you can work alone or you work together, so you're on your honor as a Fisher College student. But again, I can't monitor that. You can pick up the exam today at 4, do 5 questions, and pick it up again the next day. There's no like time limit on it in terms of like you have to complete it in an hour or you have to complete it in one sitting. As long as you start the exam before it's due and turn it in before it's due, you're all set. And I just want to make sure everyone sort of knows. Make sure you hit submit. Don't just like do it and think you've submitted. Make sure you go and hit submit because you'll get your grade right away. It'll tell you what your final score is right away. And so make sure you get your score, and that way you know it's submitted. Okay. It won't tell you what questions you got wrong right away. It will tell you the questions you got wrong and tell you the questions you got right after the fact. So Saturday at 4 o'clock after the exam is completed, you'll get a list. You'll be able to go back and look at the exam. This is what you got right. This is what you got wrong. And if you have any questions about those things, please just let me know. I'm happy to chat with you if you have any questions. But anytime you have questions along the way, just shoot me an email. I'll try my best to respond as fast as I can. I can't promise anything, Saturday before the exam is due. So, I recommend taking a peek, at least as a sort of a cursory thing to make sure you understand what's going on. But in terms of what we're covering on the exam, it's basically we're covering macromolecules, evolution, chemistry, and a little bit about how science is done. So just these four things. What we're talking about today in terms of cellular structure won't be on until the next. And now your next exam won't be up until the beginning of November. So, and just as a note, there's no homework due this week or anything like that. You just got your exam to focus on. So that's the sort of the gist of the exam. Nothing, like I said, nothing crazy. 40 questions, so you could do it in about an hour. Just because it's multiple choice, I'm just going to tell you now, easy. Like one of the things as because I can't I don't want to be the kind of professor that makes you sit down at a time and like have your camera on so I can monitor if you're cheating. I don't like that. And so to make it a challenge, because you have all the internet at your disposal, I just make it more difficult. So don't expect it to be a cakewalk. I'll say that. And just because this one is really hard, just want to remind you guys that you do get to drop your lowest exam. So if this one was really, really hard and everyone does bad, I'll break for the second one and the third one, just as an FYI. So we'll see how you guys do. But remember, don't worry too much, right? Even if you get a 50% on this exam, right, you still get 5% of your grade, right? Because each exam is worth 10% of your grade, right? So even if you bomb this exam and you bomb the second one and you bomb the third one, you could still get an A. So just don't feel too bad, okay? But again, any questions along the way, just let me know. It's an exam. I don't really have much. I don't really got much else to say about it. It's just answer the questions. So just answer those oh so fun questions. So today we're going to be jumping into, I think, my favorite topic for this semester. I like proteins a lot, but I really like talking about cellular structure. It's just like a fun topic because it's sort of like unravel. It's like this is the first time where you're unraveling sort of how you work at the cellular level, and I think that's really fun. And that's just me talking. You probably don't feel very fun, but I find it to be very fun. So let's dive into this. So cells themselves were discovered in the 1660s by Robert Hooke. And so he's the first guy, he coined the term cells. He's the first guy that observed them. But it wasn't until about 100 years later, till we had our friend Anton von Leeuwenhoek, or Anti Leeuwenhoek. He was the first person to get actual good, clear images of cells. And he wasn't too familiar with Hooke's work, but he called them animalcules. And I'd like to actually just talk about Leeuwenhoek for just a moment, because he's a really interesting guy. And so he invented these first microscopes, and they were very, very simple. So when you guys finally get back into the lab with me, maybe next semester or the semester after, you'll get to use real, like actual light microscopes. But back in the day, this is what Leeuwenhoek developed. He developed this little piece of metal attached to, basically like a bracket. Think about 1770s technology, not like modern day screws. And he had a little bit like this little like poker. That's where the specimen would go, and he had a lens. And so he had a single lens, which is in contrast to what you will be using in the lab, which are multi-lensed. And he would just mount the sample here on the little thing, and he would just look at it like this person in this image is doing. And he would draw what he saw. And so he was drawing bacteria that were rod-shaped, and he was drawing bacteria that were spiral-shaped. And he was drawing how bacteria would move. So he was doing all sorts of cool stuff. And he's best known for looking at his dental plaque. So he would, you can only imagine how gross people's mouths were back in the 1700s. But he would just take a little piece of like metal, like here. He would scrape his teeth to get all the dental plaque off, which again was probably disgusting. And then he would look at it under the microscope. And Leeuwenhoek's microscopes, again, they're particularly exciting, but his big breakthrough and his secret was his lenses, because he could make spherical lenses. And Robert Hooke, the guy who sort of discovers cells first, he didn't have spherical lenses. And spherical lenses, they don't seem like a big deal. But back in the 1700s, they were amazing. Like they were the cutting edge technology of biology. So really interesting things. And his microscopes were magnifying things about 50 to 300 times. So, you could see bacteria, you could see your own cells, you could see nucleus, but it's just sort of a coarse view. And as you can sort of see here, sort of big things. But after our friend Hooke and Leeuwenhoek, microscope-based science actually didn't really take off for another 200 years. And the reason it didn't take off, it wasn't because of Hooke. It was actually because of Leeuwenhoek. And it's kind of a jerk. There's lots of jerks in the history of science, just as an eye. You'll sort of see them as you read your textbook. But Leeuwenhoek was super proud and super paranoid about his lenses, and he didn't want to give people his lenses. So he hid his technology. He destroyed it all before he died, so no one could get his lenses. And he actually single-handedly stalled field of cell biology for 200 years, because no one could figure out how Leeuwenhoek made his lenses. So again, single-handedly, because of his just being a jerk, stalled science for a while. So kind of a how you're not supposed to do science. Science is inherently very collaborative, and Leeuwenhoek was just not collaborative at all. Now, moving on, 1830s, we had Matthias Schleiden. conducted some of the earliest types of cells, and then Theodore Schwann again a year later. But together they came up what we're going to talk about today, which is the cell theory. And so what is the cell theory? Well, it has three main components to it. All organisms are composed of cells, big or small, right? I'm cells, your cells, cats are cells, bacteria are cells, everything is cells. Cells themselves are the smallest living unit. So a cell can't get any smaller, right? You just have a cell and you can't decompose that into any other biologically functioning thing. So you just have a cell. And then cells themselves have to arise from pre-existing cells. Now, this sort of last one is an interesting one, just as a note. But this third one didn't apply to early life on the planet, right? Early life on the planet would have had to figure out a way to make cells, sort of at the first step. But I digress. But ultimately, all cells living today essentially descend from the very first living cells as a whole. And your living cells descended from an original cell that made you. And we'll talk about how that process works later in the semester. But everything is made out of cells. And cells, as you can imagine, and based on what I've told you, and I'm sure you've probably heard this, but cells are very small, right? They're microscopic. Now, there are macroscopic cells. You can physically see some cells. They're so big, you can see them with your eyes. But most cells are microscopic. And they're very, very small. And because most cells rely on the process of diffusion, right? The movement of things from a high concentration to a movement of things at low concentration to move things in and out of the cell. Now, discussed last class, things can move in and out of the cell via proteins and stuff like that. But primarily we're speaking, things need to move via diffusion. Now, diffusion itself, in terms of moving in and out of a cell, is affected by a couple different things, including surface area, so the overall surface of a cell, temperature, whether there's a concentration gradient present or not, and overall distance. And the distance we're talking about here, and the surface area we're talking about here, is again at the microscopic level. It's really sort of a weird thing to sort of think about, but that's what we're thinking about here. Now, we can look at just two types of cells here. So we have two eukaryotic cells. So we can see they both have nucleus. And so they have a cell radius. So remember, the cell radius, or the radius of anything, is just half the diameter. And the diameter is just a straight line through the middle of a circle. And so we have 1 unit and 10 units, so some arbitrary unit. Then we look at the surface area. So you calculate that with 4 pi r squared, if you remember from geometry all those years ago. You get surface area of the small one is 12.57, and the large one is 1257. So we're going up by a factor of 100, but we're only increasing the radius by a factor of 10. Now, this is where things get a little bit more crazy. We calculate the volume, so 4/3 pi r cubed. Again, geometry, not my forte, but that's the math. And we get the volume as 4.189 units cubed. And then the volume of our larger cell is 4,000 units cubed. And this is going up by a factor of 1,000. And the radius is only going up by a factor of 10. And that's an important thing. Cell size, as you sort of get bigger and bigger, the volume and the surface area expand rapidly, like really, really fast. And that's sort of an important feature of cells. As you get bigger, all the dimensions about you expand much quicker than you would imagine. And it's all based upon the math in this table. And the other sort of calculation you can look at is the surface area divided by the volume. And so the small cell has a surface area to volume ratio of 3, which is 10 times bigger than our other one, which tells you that overall, the surface area to volume ratio of our small cells stays larger. And that will become important in just a moment when we talk about it. But this area to volume ratio that has much more smaller cells does have an advantage of those that are comprised of larger cells. Because as I mentioned, as the cell size increases, its volume increases much more rapidly. And essentially, as your volume increases, and your surface area increases, it makes things harder for diffusion makes things harder for things to diffuse across. They diffuse much more slowly over larger areas. So keeping your surface area to volume ratio higher, so having more area relative to the volume, allows things to diffuse faster. as opposed to having a very low surface area to volume ratio, which means things diffuse slower. And so, as I mentioned, as cell size increases, its volume increases much more rapidly than its surface area. And so your cells can overcome this. So for instance, neurons are very long and skinny, which means they limit the amount of volume they have, but increase their surface area, which means things can diffuse faster through your neurons. Which, if you think about what your neurons are for, or just basic things like talking and walking, right? It's pretty important for things to be able to diffuse fast through your neurons, right? Because that's an instantaneous thing you need. And that, again, that's all reliant on diffusion. And so we can look at the scale of some cellular components. And I always like these graphs, but I do want to point out about this graph, it is logarithmic and it's a log of base 10. So, this mark here is 100, and then this is 10 times smaller, and then 10 times smaller, and so on and so forth. So this is a logarithmic scale. And so we have here is the human eye, so we can see things bigger than 100 meters, and as small as about, half a millimeter in size, just with our naked eye. And then we start to move into light microscopes and then eventually electron microscopes to start to see much more intact things. And so I like these little scales because, it sort of shows you your average human is, a meter and a half tall. A chicken egg is, a few centimeters. Then we move to a frog egg, and then we move to our first microscopic organism. Then we look at human eggs, and then we sort of move down the scale, right? Eventually hitting a virus, right? So this one has a virus of HIV. Also, just as a note, would be the size of COVID-19 as well. It's about 100 nanometers. And then we move down into what we're going to be talking about today, ribosomes, so cellular organelles, and we see those up here as well. And then we move down to the very, very small, where we have amino acids, so our building blocks for proteins, and hydrogen atoms all the way down at 0.1 nanometers. So it's a pretty huge scale, right? And it changes very rapidly. But cellular parts, as we're going to discuss, they are on the smaller side, right? Cells are on the smaller side. They're microscopic. Now, I just want to put this sort of scale into context. I think it's lost looking here, so I just want to put this scale into context. So the relative ranges in the size of most cells is huge. And again, going from this to this doesn't seem like a big jump. Remember, this is by a factor of 10. So it's a big jump. So in this table below, we have an amoeba, which is the thing at the bottom, so Pilomixa. It is 5 millimeters in length. And then we have the variola virus, the varicella virus, which causes chickenpox and shingles, is 100 nanometers or 10 to the minus 7 meters. So what I want you to think about is comparing this 10 to the minus 7 meters to 5 millimeters. Now, if I were to scale the virus to be the size of the mouse, how big would the protist be, this amoeba here at the bottom? So remember, the protist at 5 millimeters is 10,000 times larger than the virus. So if I scaled the virus to be the size of a mouse, how big would the protist be? Anybody have any thoughts? You could type it in the chat if you want to just throw out a guess. No one wants to try? No one wants to throw out a guess how big the protist would be if it was 10,000 times the size of a mouse? I'm not gonna lie, I'm a little disappointed. I'm pretty disappointed on this one. Anyways, if you were to scale the protist up to be 10,000 times the size of a mouse, the protist itself would be the size of the potential center. So that's 10,000 times, right? That's order of magnitude we're talking about in terms of differences in cell size and different parts of cells. It's sort of this scale. I think it gets lost, but putting it in something that you all know, that you can see the potential center from campus here. And I'm sure you guys have all seen a mouse, right? So that size difference, 10,000 times may not sound big when you're thinking about microscopic things, but putting it into sort of the context of mouse and potential, I think that becomes much more clear. So there are a lot of basic structural similarities between all organisms. So they have a nucleoid or a nucleus, and this is where the DNA itself is housed. They have a cytoplasm, which is a semi-fluid matrix of cytosol, which is basically just water with some ions in it, as well as organelles if you're in a eukaryotic organism. You have ribosomes, which is a site of protein synthesis, and they all have some sort of plasma membrane, and typically it's a phospholipid bilayer, as we discussed previously. Now, we'll start off with the most simple and definitely the best type of cells, and then we're going to talk about our prokaryotic cells, and these include our bacteria and our archaea, so two distinct domains of life here. They're microscopic, they're single cellular, but they are very different organisms. Now, prokaryotic cells, their biggest distinguishing feature from eukaryotic cells is they lack membrane-bound nuclei as well as membrane-bound organelles. Now they house their their DNA in what's called the nucleoid region. It's just a place in their cytoplasm where their DNA sits. In addition, they typically have a cell outside of their plasma membrane, though that's not always the case, but it is very common. And they do contain ribosomes, but again, no membrane-bound organelles. And so when we look at a simple prokaryotic structure, this is just our friend E. coli, very, very simple bacteria. You can see it's got its its nucleoid region. It houses its DNA. It's got its cytoplasm here in orangey color. I would say that's an orange. I guess a light orange, like a creamsicle almost. Um, and that's the cytoplasm. It's that sort of watery liquid with ions in it. And then you can see it's got ribosomes as well. And then it's got its phospholipid bilayer. And then it's got a cell wall. And it has some other things like a flagella and pili, but we're not going to talk about those right now. So this is what we're looking at. Prokaryotes, very, very simple. Very, very simple, which makes them easier to work with. But we're going to be talking about much more things today besides our friendly prokaryotes here. Now, in terms of the cell wall of bacteria, most bacteria are encased by some sort of cell wall, and it's typically pretty strong. It's designed to give them a pretty heavy amount of structural rigidity. It's actually kind of an interesting structure. If you've ever seen a chain link fence, that's what a bacterial cell wall looks like. It's just sort of it's sugary and proteinaceous, and it looks like a chain link fence. It's actually kind of cool. But the primary component of a cell wall is peptidoglycan, and it's just a sugar. There are cell walls in plants, fungi, and protists, but they're made out of different compounds outside of peptidoglycan. So I don't want you to get confused that cell walls are just only found in bacteria. You can find them in eukaryotic organisms as well. They do all sorts of things. They help maintain shape. They protect the cell. They prevent excess uptake of nutrients and water and stuff like that. And they also make them less or more susceptible to antibiotics. So if you have a very thick cell wall, you're less susceptible to antibiotics, just as a note. So many of the most clinically important bacteria out there have a very, very thick cell wall that protects them from a lot of our antibiotics. And I'll just make a note that archaea also lack peptidoglycan. Our flagella, they're present in some prokaryotes, and some have multiple copies, some have one copy, some have weird orientations, but they're used for locomotion, i.e. movement. And so this is what a flagella looks like. This is its sort of overall structure. And what it does is just sort of whips like a tail. It spins like a corkscrew, as you sort of see here. And again, this just powers the bacteria through. This is very much how sperm swim. Sperm don't like have a tail like a dolphin swimming. They corkscrew it, very much like a bacteria does. So that's our prokaryotes. Very, very simple, which I think is which is nice. But what we're going to spend most of the class talking about is our eukaryotic cells. So these include plants, fungi, mammals, invertebrates, basically anything big that you can, think of. Now, prokaryotes have membrane-bound nucleus. They're overall more complex, and they have what's called compartmentalization. And when we're thinking about what compartmentalization is, it's just the presence of membrane-bound organelles and an endomembrane system, both of which we're going to be talking about. And in addition to this, our eukaryotic cells also have a cytoskeleton, which is we use for support and maintenance of cell structure. So before we sort of dive into some of these organelles, let's talk about the differences between the two major classes of cells, and these are going to be plants and animal cells. And so plants and animal cells are basically identical, but they do have a few key differences. So they have plasma membranes, they have most of the same organelles, they have phospholipid bilayers, they have a cytoplasm. Most of the stuff is similar, but plant cells have a few extra components typically not present in other eukaryotic cells, including a cell wall, chloroplasts, as well as vacuoles. And so we'll talk about these today. But just to sort of show you what an animal cell looks like, and then a cell, this is what an animal cell looks like. It's typically circular in shape. You can see all the different organelles. And we're going to talk about all these: the nucleus, the mitochondria, endoplasmic reticulum, the centrioles. We're going to talk about all these different organelles soon enough. And that's in contrast to our plant cells, right? Very different overall shape. And again, you can see that rigid cell wall, and you can see the vacuole here, as well as chloroplast. So very similar, but plants have a little bit more speciality to them than animals do. But I will also make a mention that animal cells also have this organelle here, which is called a centriole. And that's involved with cellular division. So we'll we'll talk about that one later. Later in this semester, I should say. So let's start with the nucleus because this is sort of the brain of your cell. And so the term organelle, which I've been using, just describes any of these membrane-bound compartments. And you sort of think about an organelle as like a mini organ. So when you look at organelle, it actually translates to mini organ. So you can think about all the things we're going to talk about as being like distinct organs in your cell, and right, and the cell being like a system, very much like your body is, right? You can think about your brain of your body is the nucleus, right? And there's a lot of parallels between your organs and the organelles inside each one of your cells. But the nucleus itself is the repository of genetic information, so it houses all your DNA. Most eukaryotic cells possess a single nucleus, though there are really cool eukaryotic cells out there that have multiple nuclei, it's a pretty interesting group of organisms, but maybe we'll talk about it next semester in Bio 2 because it's much more nature-y. They also have a nucleolus. And the nucleolus, as you'll see, is just sort of attached to the nucleus itself, and it's a region where ribosomal RNA synthesis takes place, and we'll discuss that very shortly. The nucleus also has a special membrane, as all organelles do, and it's called the nuclear envelope, and it is comprised of two phospholipid bilayers, and it sort of spread throughout the phospholipid layer is nuclear pores that control movement in or out of the cell. And just as a note, in most eukaryotes, the DNA itself is divided into multiple linear chromosomes. And we'll talk a lot more about chromosomes when we get into the nitty-gritties of DNA. But we can just look at this organelle. So this is our nucleus in our sort of animal cell depiction here, and you can see our nucleus here. Nothing really crazy, but you can see the double lip phospholipid layer here with the nuclear pores, and you can sort of sort of a zoomed in picture of what a nuclear pore actually physically looks like. So you see there's an inner membrane and the outer membrane, and then you have the nuclear pore, which is basically comprised of mostly proteins as well as some lipids as well. And just to sort of show you, this is what it looks like when you actually look at it under a microscope, so not like a cartoonized version. And then you can also see the nucleolus kind of smack dab in the middle there attached to the nucleus. So very simple structure, but and its primary advantage just mostly for housing your DNA. We'll talk about sort of more of the roles that the nucleus do as we sort of learn more about DNA and transcription and stuff like that. But just as a sort of introduction to the nucleus, that's what it looks like. So another organelle that we're going to discuss, and this one you find in bacteria and archaea as well, and these are ribosomes. And ribosomes are the site of protein synthesis. And they have a sort of an interesting structure that I'll show you. They kind of look like a hamburger bun, or maybe like, I don't know. I always think a hamburger bun, but I can't think of a better analogy. Maybe you guys can think of a better analogy when you see a picture of a ribosome. But ribosomes are basically just protein structures. Yep. Oh, did you have a question or? Okay. Now the ribosomes themselves are a complex of ribosomal RNA, as well as several different proteins. And the ribosomes themselves can be either floating around free in the cytoplasm, or they can be associated with internal membranes. And so let's look at the ribosome, what it looks like. Again, I think it looks like a hamburger bun. I don't know, maybe someone could come up with a better analogy for what that looks like, but I'm gonna go with hamburger bun. Or maybe a roll. I guess a roll would also work. But it has two components. It has a large subunit and a small subunit. And the large subunit and the small subunit are about 40% protein and about 60% ribosomal RNA. We haven't really talked too much about nucleic acids, but ribosomal RNA is a special type of RNA. It's structural in nature, and it forms the part of our ribosome. So again, ribosomes themselves, 40% protein, 60% ribosomal RNA. And again, this is the site of protein synthesis. And we'll get into that when we start to talk about transcription and translation as the semester progresses. But this is our ribosomes, the hamburger bun that's making all your proteins. And just as a note, again, I mentioned they can be found floating free in the cytoplasm, and that's where they are in bacteria as well. But you also notice that we have an organelle here, which we're going to talk about very shortly, that's covered in ribosomes. So it can be either membrane or organelle attached, or just hanging around in the cytoplasm. And typically speaking, if it's in the cytoplasm, it's doing something in the cytoplasm. So it's producing a protein for the cytoplasm. But if it's on organelle, it's producing a protein for something else that's not related to the cytoplasm itself, just as a note. So before we move on, we're going to discuss the endomembrane system, because we need to understand the endomembrane system before we talk about the next series of organelles. Now, very much like you can move things around your body via your bloodstream, cells also need to move things inside the cell, and they do it by the endomembrane system. And the endomembrane system relies on a series of membranes throughout the cytoplasm, and ultimately it divides your cells into multiple compartments where different cellular functions occur. Now, these different compartments are just the organelles. That's all. So your cells, as you see in this picture, they have different membrane-bound compartments that are just organelles. And so the endomembrane system just describes the process of how things move from organelle to organelle to organelle to potentially outside of the cell. And this is a huge, I should also mention, this is a huge distinction between eukaryotes and prokaryotes. This endomembrane system only exists in eukaryotic cells. Our bacteria and archaea just don't do this. So to end this system, we're going to talk about the very first organelle, which is the or the RER, the rough endoplasmic reticulum, as well as its counterpart, the SUR, the smooth endoplasmic reticulum. Now, these two membrane-bound organelles have two distinct functions. Now, the rough endoplasmic reticulum is really only different from the smooth one in the sense that the rough endoplasmic reticulum has ribosomes attached to it, giving it essentially a rough appearance. So you can see that in this diagram here, right? They look very similar, right? They're just membrane-bound organelles, but one has ribosomes and the other doesn't. Now, the rough endoplasmic reticulum, as you can imagine, because it's covered in ribosomes, it is a huge site for protein synthesis. And also, in terms of where those proteins end up going, they're typically sent to the plasma membrane to be excreted from the cell, or they're sent to other parts of your cell, potentially the mitochondria or the lysosome, things like that. So that's our rough endoplasmic reticulum. And our smooth endoplasmic reticulum typically has no, but it sometimes can have some ribosomes attached to it. And it has a variety of functions. And so its most common function that it does is it synthesizes lipids. So fats, whether it's for storage or whether it's for your membranes or something like that. It can also store calcium. So that's a pretty big deal for muscle cells, which rely on the exchange of ions such as calcium to contract your muscle. And they're also could be involved in detoxification. But ultimately, the ratio of the rough and smooth together depends on your cell's function. So cells that are involved heavily in making proteins or enzymes, such as your liver, have a lot of rough endoplasmic reticulum. Those that have much more smooth endoplasmic reticulum are typically involved with the production of detoxification. So your kidney cells have lots of smooth endoplasmic reticulum, as does your pancreatic cells. So it just depends. And different cells in your body, your liver, your brain, your heart, they all have different proportions of organelles, which I think is a sort of tie. Not that I think it ties directly into their overall function. But in terms of what this looks like, as I mentioned, the rough and the smooth, they're just membrane bound. You'll notice that the rough endoplasmic reticulum does border the nucleus. So, and we'll talk about why that is when we get to, transcription and translation. And then our friend, the smooth endoplasmic reticulum, typically borders the rough endoplasmic reticulum. And if you want to sort of see what that looks like microscopically, they just kind of look like a stack of deli meats. That's the sort of the way I think about it. For at least the rough endoplasmic reticulum, looks like you just took a bunch of deli meats and kind of stacked them on top of each other. And the rough endoplasmic reticulum, I always thought they kind of look like coral, that structure of coral. But again, that's just how I see things. So that's basically the site of synthesis, right? Fats or proteins or anything, it starts with the endoplasmic reticulum. But for things to be transported from point A to point B, we have to send them to the Golgi apparatus. Now, the Golgi apparatus is a flattened sac of interconnected membranes, which are called Golgi bodies. And now these functions as a packaging and distribution essentially center. So you think about like UPS or USPS or FedEx. That's basically what the Golgi apparatus is. It packs things in vesicles, which are just membranes, tags them with the signal of where they're going to go, and sends them to their destination. And it has some directionality. So it has a face and a transface. And let's look at what this looks like. So this is the Golgi apparatus. You see it's sort of it's near the reticulum, but it's sort of near it's near the cellular membrane itself. And again, it just looks like a bunch of these individual membranes. These are what are the Golgi bodies. But and this is what it looks like microscopically. And it's always important to look at the scales here because this is only 10 microns wide. It's not very big. And it does have directionality. So it has a cis face, and the cis face always faces the nucleus. And then it has a trans face, which always faces the not the cytoplasm. Well, the cytoplasm or the membrane itself. But ultimately, the idea being is once you get a protein or a lipid or whatever, you send it to the Golgi, and the Golgi packages it in one of these transport vesicles. And all that is just a piece of membrane of the Golgi. That's really all it is, which is kind of nice. You're just butting off little pieces of this membrane, which means you have to consistently resynthesize more membrane for the Golgi, but that's just par for the course and how this works. Now let's sort of pull these three organelles together and look at this nice diagram. So we have some process that happens in the nucleus. We want to make a protein, so we have to access our DNA. We send the message to the endoplasmic reticulum, where it's synthesized into a protein. Well, once we make that protein, well, we need to do something with that protein. So what we do is we send the protein in a vesicle. So we basically butt off a piece of the endoplasmic reticulum, and this could be the same thing for the smooth endoplasmic reticulum. But you send it off in a little vesicle. It's just a piece of the membrane, and you send it across to the Golgi apparatus, where it is one of two things happens. It's two things that happen to it. It is essentially fuses to the Golgi and passes from that cis face all the way to the trans face. It just basically think about it like just melting through the Golgi apparatus itself. But as it passes through the Golgi apparatus itself, it's tagged with what we call a surface recognition protein, or an SRP, that tells it where to go. And then once it exits the trans face of the Golgi, it then gets sent to where it needs to go. And so that can be to outside to the cellular membrane to be excreted, can be to the mitochondria, could be to any of the other organelles present. But that's the flow of information. You start off with the endoplasmic reticulum, either smooth or rough. It gets transferred via vesicle to the Golgi, where it's tagged and sent to wherever it needs to go. Again, you can think, if you want to put this as an analogy of like, real world things, like you could pretend you have a package, and so you take your package, you hop into your car, and your car would be one of your vesicles, and you drive to the post office, and then you give that package to the post office, and the post office goes, hey, You got this zip code on it, you have this address, cool. I'm going to put it on my truck and I'm going to send it where it needs to go. And that's the flow of information we have here. Very, very similar to how you ship a package, which is nice. And it is a very simple system. Now, it's simple for our purposes. It does get a lot more complex. You know, the molecular biology behind it is pretty complex, but at its surface level, what I want you to remember is that, again, this flow of information. The sort of the key for this sort of lecture, and many of the lectures we talk about, is big picture stuff. Because as you sort of progress in your career at Fisher, you'll learn more than nitty gritties, but we need to understand the big sort of overall picture first. So next up on our train of organelles is the lysozyme, and these are membrane-bound digestive vesicles. And these are basically organelles that arise from the Golgi apparatus. So something gets sent to the Golgi apparatus, and and it basically is marked for destruction. Well, the vesicle of the Golgi apparatus then becomes a lysosome. Now, the lysosomes contain enzymes that catalyze the breakdown of macromolecules, so proteins, fats, carbs, lipids, things like that. And ultimately, to do this, they typically fuse with the target to initiate a breakdown. And they're used to recycle any number of different things, including old organelles, foreign cells, or potentially foreign matter that the cell has engulfed. So the idea being is we start with that flow of information and we synthesize some sort of protein. In this case, it's a protein that it's going to be used as a lysosome. And we synthesize that protein at the rough endoplasmic reticulum. It gets sent to the Golgi, where it's packaged again, and it gets repackaged in the membrane of the Golgi apparatus. Well, if this protein is now involved with the digestion of a organelle, such as the mitochondria here, that lysosome essentially travels through the cytoplasm, fuses with the damaged organelles, engulfing it, like literally like engulfing it, and then it starts to use its enzymes to break it down. Or you could imagine that maybe the cell has uptaken something from the environment. Maybe it's a bacteria, maybe it's food. And what ends up happening is the lysosome binds with that food, uses all its enzymes and chemistry to digest those products. But that's ultimately what you're thinking about for the lysosome. Comes from the Golgi, but whatever it's digesting, it's doing it by fusing with it and engulfing it. So it's kind of like Pac-Man. A lot of the stuff we talk about is kind of like Pac-Man because everything just kind of likes to nomp on one another. So moving on for the lysosome, there are what we call microbodies, and the one we're going to talk about today is just one class of these enzyme-bearing membrane-enclosed vesicles, and this is the perioxisome. And now perioxisome contains numerous enzymes involved with the oxidation of fatty acids, so your lipids and things like that. And one of the byproducts of this is hydrogen peroxide. But hydrogen peroxide is what we call a reactive oxygen species, and it is very, very destructive. And so by doing this process inside of a perioxisome, you prevent hydrogen peroxide from getting outside of the perioxisome and damaging the cell around it. Now, the perioxisomes also contain an enzyme called catalase, which converts very toxic hydrogen peroxide to harmless oxygen and water. So if you've ever used hydrogen peroxide to clean a wound, like a cut on your skin or something like that, and it bubbles, what's happening is that the catalase enzyme on your skin is breaking down the hydrogen peroxide, and the bubbles you're seeing when you clean yourself with hydrogen peroxide is oxygen being produced. So fun fact in case you ever wanted to know why hydrogen peroxide bubbles when you clean a cut with it, which is, just fun. And in terms of perioxisome structure, it's not particularly very exciting. They're generally just kind of hanging around, either attached to the cellular membrane or hanging around the cytoplasm. They're circular. Nothing really to them in terms of overall structure, but they're not particularly very big. You can see the overall size here is like 1 micron wide. It's not very big overall. So something that only plants have and animal cells lack is vacuoles, and these are membrane-bound structures, as all organelles are, and they do have varying functions depending on their cell type. And so there's a couple different types. So there's the central vacuole in plants, there's storage vacuoles in plants, and then there's contractile vacuoles in plants, fungi and protists. Now they all do very, very different things. The central vacuole in plants is typically involved with the storage of water. Storage vacuoles are typically involved with the storage of starch, which is a polysaccharide, very, very large polysaccharide. And then contractile vacuoles are typically involved with contraction of cells. And that's typically involved with movement of these two. But we won't really focus on these last ones. We'll just sort of focus on these two for plants. And we can look at what this looks like. And so we see we have the nucleus of the plant, we have the chloroplast, and we have that central vacuole here. And you can see the central vacuole is just chock full of water. And it's kind of an interesting thing about the central vacuole is it can essentially fill up to 90% of the volume in a plant cell. So a vacuole can count for most of the volume of a plant cell, and it counts for most of the volume when it's filled with water. And this sort of interesting thing is, as you fill and fill a central vacuole with more and more water, it causes the plant cells to swell. They get what's called turgor pressure. And the interesting thing about turgor pressure is it makes the plant cells rigid and tough. And so when a plant is healthy, it has very high turgor pressure, and the plant stands up straight. When a plant is low on water, the turgor pressure goes down, causing the plant cells to become less rigid, and the plant starts to wilt. So if you ever wonder if a plant was wilting, at the cellular level, it's because its central vacuole is not full. So anytime you see wilting plants, the most common thing to do is just water them, and they'll typically come back to life by, again, filling their central vacuole, reestablishing turgor pressure, allowing them to grow straight up. So for any of you gardeners out there or people who aren't very good at gardening, that's a pretty good tip. So moving on from our vacuoles, we're going to talk about my favorite organelle, which is the mitochondria, and this is something that's found in all eukaryotic cells, plants, fungi, animals, doesn't matter. It has multiple structure to it, so it has an outer membrane, an intermembrane space, and then it has a second inner membrane shaped into folds that are called cristae, and then inside the cristae it has a matrix. And now mitochondria has colloquially being called the powerhouse of the cell. And I learned recently that's a meme. I didn't know that till recently. I kind of think that's funny. But it is the powerhouse of the cell. It's the site of cellular respiration. Now, mitochondria are cool. They have their own DNA. And one of the kind of cool things about mitochondria is that we do know that they are their ancestor in terms of before eukaryotes existed was actually free-living bacteria. And so they look like bacteria, they behave like bacteria, they actually divide all on their own, and when your cells are dividing, and they have their own DNA. So mitochondria are a wild and wacky place, but again, they are the site of cellular respiration, which is a topic we'll discuss in a few lectures. And when I say cellular respiration, I just mean how your cells generate energy from sugars and fats and proteins. But again, this is the cell. It's sort of it's bean shaped almost, and again, it has that outer membrane, and then it has the inner membrane space, and then it has that second inner membrane here, which forms this weird sort of overall shape. Now, inside this inner membrane, we have the matrix, and in between the folds of our inner membrane, we have the cristae, or sorry, the crista in this one, but it can also be cristae. And so this is our mitochondria, and its structure will become much more clear when we actually start to talk about the process of cellular respiration and why there's all these folds and these spaces and stuff like that. But the other thing to note about this, I mentioned it has its own DNA, but you also notice that the mitochondria also has its own ribosomes. So the mitochondria itself is it's sort of a one-man army in terms of what it does. It's got everything, which I think is pretty fun. The plant counterpart to mitochondria, though plants do have mitochondria, is the chloroplast, and this is an organelle present in plants as well as other photosynthetic eukaryotes, and very much like our mitochondria, it is a multiple membrane system, But in this case of the chloroplast, it has two membranes that surround it. And our chloroplasts contain chlorophyll, which is a light capturing pigment, which we'll talk about later on in the semester when we discuss photosynthesis. But it also has these things called thylakoids, which these are these membranous sacs. And as you stack thylakoids, multi high, you start to form what are called grana. And also, like our mitochondria, they have their own DNA, and they also have their own ribosomes. Now, the ancestor for chloroplasts actually turns out to be photosynthetic bacteria, just as a note. So let's look at the structure here. We have those two membrane forming the outer layers here, and then we have our thylakoid discs, and inside those thylakoid discs, we have some internal structure here. So inside the overall structure, we have grana, which are the sort of little fibers here, and then we have roma as well. But as we start to stack these up together, they form that granum. Just as a note about sort of what's going on here, all this green color that we're seeing and why plant cells are green and plants look green and leaves are green, it's because of the presence of chlorophyll, which is a pigment that captures light, and chlorophyll is a green pigment, which is why chloroplasts appear green. So just as a note. So, but as I've sort of been alluding to about how we get the chloroplasts and how we get the mitochondria, we get them through what we call the endosymbiotic theory. Now, the endosymbiotic theory is a really interesting theory. It was proposed by Lynn Margulis. She's at, or at least she used to be at UMass Amherst. I believe she's retired now. She's a really if you're interested in anything, learning about awesome women in science, Lynn Margulis is an awesome woman in science because this theory that she proposed in the 90s, she was ridiculed for it, but she stuck to her guns, and as our techniques got fancier and fancier, she was proven essentially right. So she's a really awesome woman if you want to sort of read up about her. Her name is Lynn Margulis. Very, very cool woman. But her theory, which is pretty heavily accepted across the scientific community, proposes that present-day eukaryotic organelles evolved by symbiosis between two free-living cells. Now, let's break that apart. What does symbiosis mean? Well, symbiosis means just a relationship between two organisms. So they are living together, potentially benefiting one another. That's what a symbiosis is, right? And so the idea being, the way we get our organelles is that one cell, a ancestral prokaryote, was engulfed by a larger cell, which then became the precursor to our modern day organelles. And so as I've mentioned with the mitochondria, we believe it was a free-living heterotrophic bacteria, so a bacteria that liked to break down sugars for its metabolism. That was the ancestor for the mitochondria that was engulfed by a bigger organism. But as they lived together over time, we had the development of the organelles. And then the chloroplasts was a free-living, what we call a cyanobacteria, that was engulfed by a larger cell, which eventually over time and through symbiosis became a chloroplast. And so there is a lot of sort of evidence that supports this theory. And as you come up in the ranks at Fisher and you take more courses, you'll learn more about all the evidence. But just as a note, most of our organelles we can trace back to some sort of bacteria. And so we do know that the nucleus likely came from a symbiosis with an archaea. Mitochondria came from a symbiosis with some sort of bacteria. And our chloroplast came from some sort of symbiosis with a cyanobacteria. So we do know all these things existed, but it started with some larger bacteria being able to engulf many smaller bacteria. And that's the idea. And if you can engulf them and they're living inside you and you don't kill them because there's some benefit to it, evolution will kick in and start to change things overall. So it's a really kind of fun really, really fun theory that I wish we could dive more into, but I just wanted to give you some, a couple threads as to how organelles came to be, because we saw the original bacterial cell no organelles. But then we go up to the eukaryotic cells, we have organelles. And so how we got from no organelles to organelles is via the endosymbiotic theory. So if you want to learn more about that, I'd be happy to give you some more information about it. I think, like I said, I think it's very fascinating. But for the purposes of this class, again, symbiosis is how we get our organelles. Next up, we're going to talk about our cytoskeleton, and this is a supporting structure. Now, this is something we have, but bacteria also have a primitive cytoskeleton, as do archaea. And actually, as a kind of a fun note, the cytoskeleton that we have actually is very similar to the bacterial cytoskeleton. So the proteins and the structures are very bacterial-like, just as a note. Now, the cytoskeleton, you kind of got to think about it as like a net-like structure. It's a network of fibrous proteins that's found across all eukaryotes that basically like acts as like a mesh around the cell, but like inside the plasma membrane. And it does this to support the shape of the cell and keeps the organelles bound to fixed locations. Now, every organelle is physically attached to that cytoskeleton. Now, there are three types of fibers that make up our cytoskeleton, and they vary in their abundance depending on what type of cells you are looking at. And so we have microfilaments, which are called actin filaments, and these are two protein chains loosely twined together. You can think about it like rope. And this is involved with contraction or crawling, crawling on your hands and knees, or pinching. So thinking about this, muscles have lots and lots of actin. So muscles, as they need to contract, they have lots and lots of actin. Next up is microtubules, and these are very, very large. They're composed of what we call dimers of alpha and beta tubulin, and these are directly involved with the movement of things in and out of a cell. So when we talked about how the Golgi apparatus was sending things to the membrane, or sending a perioxisome or a lysosome to digest something, what they were doing is they were attaching to these microtubules to move on overall. And then finally, we have intermediate filaments. And these are, as you can imagine by their name, sort of an in-between between actin and microtubules. And they are very, very stable overall. And just as a note, these two ones up here, they do break and reform all the time. So they're not static. They do break and reform. So let's look at what these look like. So we have our actin, which again is like a kind of a twisted rope. Again, this would be part of your muscles. We have microtubules, and then intermediate filaments. And so remember, the microtubules are sort of how your organelles are hopping around your cells. But just to sort of see what they look like, we have the two plasma membranes here, and you can see we have our actin filaments. They act like a kind of mesh, right? They give that contractile ness to it. You can imagine like those actin fibers in the cell could be essentially contracted like like foam, almost like, right? That's sort of the properties that give the cell that flexibility. They kind of parallel, very much like the intermediate filaments do as well. And so they sort of how they interact. And so cytoskeleton's sort of all over the physical cell itself. It's not just like one overall part here. But again, you can also see that our organelles here, so the mitochondria and the rough endoplasmic reticulum here, are bound to the cytoskeleton, so they stay physically in place. So sort of moving on, we have a region of our animal cells, which are called centrosomes. They're also called centrioles. And these are our microtubule organizing centers. And we'll talk about these much, much more in depth later in the semester when we talk about cellular division, but just wanted to just put them forth, these centrioles. This is what they look like. You find them here. But again, just as a mention of what they are. Only found in animal cells, but they're involved with lots and lots of processes. And we're going to talk about them much more in depth when we talk about how cells divide overall. So we talked about how bacteria can move. Let's talk about how cells move. Now, your cells don't move. Sperm move, and your blood cells move, but sperm swim like bacteria, and your blood cells are carried through, your blood, right? They're the pressurized movement of liquid. But some cells, some eukaryotic cells, can move. And cellular movement for a eukaryote is involvement of the movement of the cytoskeleton. And cells essentially kind of crawl. Eukaryotic cells are very big, so it's hard for them to swim, but they sort of crawl, right? They sort of drag themselves across the surface. So if you picture kind of like a slug moving, it just kind of drags itself. That's kind of how your cells, not your cells, but eukaryotic cells that have the capacity to move. Now, that's how the vast majority of eukaryotic cells move, but some eukaryotes have flagella and other structures called cilia, which are just microtubules. Now, let me show you what that looks like. So we have a flagella here. It's very, very different than a bacterial flagella, just as a note. And ciliae also have a very similar structures as well. But you can see it's just a bunch of microtubules just bound together, and they're held together with a central core here, which is called the central microtubule pair. But it's a bunch of dual, what we call, dimers, so two tubules, so two microtubule dimers sitting with one another. And very much like the bacterial flagella, it either rotates like a sort of a corkscrew, like a propeller, or it kind of flaps in the air like, you think of like a tail. So, and then sort of one of our last stops here on the overall structure of the cell is going to be the eukaryotic cell wall. Now, the eukaryotic cell wall is present not in animal cells, so we don't have a cell wall. Cats don't have a cell wall, but it is found in our plants. It's also found in fungi, as well as many species of protists. Now, the structural components of cell walls vary depending on which organisms we're talking about. And they are very, very distinct from the bacterial and archaeal ones we discussed earlier in the lecture, both chemically as well as structurally. Now, plants and protists, they make their cell walls out of cellulose. And cellulose is a really important molecule. It's just it's carbohydrate. Fungi make their cell walls out of a carbohydrate called chitin. And that's again another carbohydrate. And then finally, I'll just make mention that plant cells sometimes also have two cell walls. They don't just have one cell wall. They sometimes have a primary and then a secondary cell wall. So, but again, we're looking mostly at cellulose and chitin here as sugars that make up the structure of our cell walls. Now, we could look at what this looks like. So we have a single plant cell here, and it's surrounded by a bunch of other plant cells, and you can see the organelles of the plant cell. But what you can also notice is that you can see the secondary cell wall, and then you can see the primary cell wall of the plants. And sort of a zoom in picture here. Again, you see that secondary cell wall, you see the plasma membrane here, and then you see that primary cell wall out here. You'll notice that the plants are very, very sort of tightly bound together. They're essentially like stuck on one another with those primary cell walls. So they're very, very tightly bound with one another. But there are places where they just can't fit in together perfectly. They have this little space called what's called the middle lamella, and just sort of this empty space in between plants. Excuse me, it's been a long day. Now, for our animal cells, they lack this sort of tightness between them, right? They don't have the cell walls binding one another. They don't have that middle lamella. They have what's called the extracellular matrix, or the ECM for short. Now, what animal cells do is they secrete this elaborate mixture of proteins and other sugars and things like that into the space around them. And some of these proteins may be proteins you've heard of, such as collagen. And what they do is they essentially form this kind of like slimy layer around your cells. Essentially just gives your cells this protective layer. And it has these things called integrins, which link the extracellular matrix to your own cell's cytoskeleton. And depending on what your extracellular matrix looks like, it will influence your overall cell's behavior. And this last point's not too important, but I figured I'd bring it up. So let's look at what we're going on here. So we have the cytoplasm, we have the cellular membrane, and then we have this extracellular matrix here, or that ECM. Now you can see we have the actin filaments inside your cell. And then we have these integrins, which are just transmembrane proteins. And what you'll notice is that they bind to your your extracellular matrix, keeping those proteins and those sugars that you've secreted to form that protective layer attach to your cells so that that extracellular matrix doesn't just kind of fly away. And the thickness and the composition of this extracellular matrix does vary depending on what type of cells you're looking at. So your liver cells have a very different extracellular matrix than your nervous cells and your kidney cells and so on and so forth. So similar compounds, but different organization, different thickness as well. So this is just a really handy table that has all the different components we talked about: cell walls, membranes, flagella, mitochondria, so on and so forth, and whether they're found in prokaryotes, animals, and plant cells. So this is just a sort of a handy dandy table that will tell you, oh, if I have a ribosome, oh, I know that's present in all three. Oh, I have centrioles, I know that's found only in animal cells, but not plants and bacteria. So sort of just a handy dandy table for you from our friends at McGraw-Hill that I like to sort of stick here. Now, that's the basic structure of a cell. There's a lot more that goes into this, but what we're concerned here mostly is the sort of the overall basic structure, right? And understanding how they're connected together through that um, transmembrane system. I just lost the word for it. But how they're connected for one another. That's the sort of the big things. What are the basic functions of these organelles? How do they move things in between them? And what are they doing? That's sort of the meat and bones here. Not the super duper specifics, but the overall big picture. That's what we're really concerned about. So we're basically going to talk a couple of a few more slides about cell-to-cell interactions. And so cells have an identity, and your liver cells are distinct from your kidney cells, they're distinct from your brain, and so on and so forth. So all cells have an identity, and the way your cells are held together does vary. Now, typically what gives your cells identity is most commonly a protein that are just sort of hanging on the outside of your cells, but sometimes it can also be sugars as well, but it's mostly proteins. Um, but the surface proteins give a cell identity and allows them to make contact and read one another and react to the presence. So if your liver cell reads another liver cell, it says, hey, we're both liver cells. Let's hang out and do livery things together. But if your liver cell came in contact with a kidney cell, he would go, whoa, you're a kidney cell. I can't work with you. And you shouldn't be here, or I shouldn't be here. So it does give them identity, allows them to interact, and allows them to work together. And there are a couple different types. So we have what are called glycolipids, and these are tissue-specific cell surface markers. And then we have what's called our MHC, or major histone complex proteins, which are recognition of self and non-selves by the immune system. So this is our cell-to-cell interactions. It's based upon knowing who is who. And again, it's all related to just proteins. You don't need to remember the specifics here. You just need to remember that I know I'm a liver cell because of the proteins. Now, pretend we have two cells that they know they're liver cells. Well, if they're both liver cells, they're going to be doing a similar function, and they're going to want to bind to one another. And so they can do that through one of these three mechanisms. So they can form what are called adhesive junctions, which are mechanical attachments to the cytoskeletons of neighboring cells, or potentially to the extracellular matrix. They can form septate or tight junctions, which are connections of the plasma membranes to adjacent cells. And I'll show you what these look like in just a moment. Or we can have what are called communication junctions, which are chemical or electrical signals, channels that allow chemical or electrical signals to pass directly from one cell to another. So let's see what these look like. So we have all three here. We have tight junctions, we have adhesive junctions, which are also called desmosomes. And then we have a communicative junction. So we're going to start off with a tight junction, and this is what we would see for your skin cells, just as a note, but they can be found a lot all over the place, just as an FYI. But these tight junctions essentially allow a super duper tight, watertight seal to form between your cells. Now you can imagine this is important for your skin, right? Because your skin needs to be waterproof, right? So having these very, very tight junctions creates a watertight between the two. Our adhesive junctions are a bit different. You got to think of your adhesive junctions kind of like Velcro, and you can sort of see that here. It kind of looks like just Velcro together. But ultimately, you'll notice from this diagram, it's connecting um, filaments, so parts of the cytoskeleton, directly to one another between these two cells. Um, and then finally, we have the communicative junction, which sorry about that. The internet in my office just sort of dropped. I don't know where it dropped. Did everybody hear me talk about adhesive junctions? Or do I have to start over on this slide? Okay. So I have to start over on this slide? Okay. So adhesive junctions, okay, I'm good. Um, okay, so I'll just talk about the final one, which is communicative junctions. And these, again, look like someone took a pencil and kind of stabbed it through the cell membranes between them. And this is for direct communication between cells that are neighboring one another. And so you see these pretty commonly throughout your body. They're really common in like organs, and they're really, really common in your neurons as well, because they allow things to pass directly through, right? So you can imagine if I have a electrical signal that needs to make it from one neuron to the next, well, having this could allow me to potentially send things directly through one of those channels, essentially instantaneously. So different types of junctions that hold our cells together overall. And then our plants have a very specialized type of junction between them, which we call plasmodesmata. And these are specialized openings in their cell walls. And basically what they do is they connect the cytoplasm, sometimes the endoplasmic reticulum as well. And they do have um, a very similar function to the adhesive junctions in our cells. Now, again, it's just a direct connection opening between the two cell walls, directly connecting either the cytoplasm and sometimes the smooth or rough endoplasmic reticulum. It just depends. Again, it allows direct transport of proteins across these cell walls, because cell walls in plants are super duper tough structures. So it's kind of hard to move things in and out of a plant. So that's a tour of our cell. That's a tour of a plant cell. Those are all the major structures. That's how they connect. It's sort of the backbone of a cell. Now we're going to talk about specific organelles coming up in the semester. So we're going to talk about the chloroplast, we're going to talk about the mitochondria, but we're going to talk about specific things later on. The point of today's class was to give us a really broad introduction to what the heck is happening at that cellular level. And so what we're going to spend the rest of our time together today is we're going to break off into groups, and we're going to do a little exercise, very much like we did before I got sick, which wasn't fun. So if you pop on over to the weekly modules folder, and you go down to week 5, we're on week 5, which is just kind of crazy. You'll notice whenever the school Wi-Fi decides slow, we have an in-class activity together. Now this in-class activity is, it's got some background information here. This shouldn't be here. It has some background information on it, so how the activity works. There's slides, there's some background knowledge for you, organelles and illness, and this is the physical activity. And there's also this also shouldn't be here. Anyways, also some background knowledge for you to read as well. So this is just a website link. Now the activity itself is pretty straightforward. And so if you open her up, man, the Wi-Fi in school is so slow today. I'm surprised I could even talk to you guys. It's been bad today. Then again, it was bad. It was bad last week too. Wow. Get rid of my video. All right. So the activity is you're pretending you're a part of a research team that specializes in diagnosing different disorders. But ultimately, what you're going to be doing is I'm going to break you guys off into your groups. We'll just do random groups this time. And one person, and there's 4 scenarios. So we have 4 different potential illnesses. And so you'll be broken up into groups of 4, and you're presented with a couple different things. Now, oh, come on. You have patient history, so it tells you about the patient. It's what the patient is experiencing. So this one is the 7-year-old male, fatigue, muscle weaknesses, low endurance, loss of coordination, and progressive difficulty walking. You get a physical examination, and you get some laboratory results. And you're trying to identify, based upon these tests, what organelle is malfunctioning and why. So in this case, we have cells that have abnormal shape, cell division is abnormal, impaired intercellular materials. Well, based on what we learned in this previous class, you want to try to figure out what potential organelle is misfunctioning. Now, there's a whole class of diseases out there that are associated with misfunctioning organelles. There's tons of them out there. And so your job today as your group is to identify which of the 4 organelles which organelles is responsible for causing the symptoms in each 4 of these cases. Now, it tells you to do as facilitator. You don't have to do that if you don't want to. This would just be like a leader for each one of these. But you want to try to identify, based on the symptoms, based upon the laboratory results, what essentially what organelle is misfiring to cause this disease. And so I know a lot of you are interested in medical science, so I thought this would be a pretty fun activity to sort of relate to you the cellular level processes and how they can actually cause meaningful diseases, because there are, again, tons of diseases out there that are caused by misfunctioning organelles. Now, if you want to read a little bit more of some of those diseases, I gave you some slides here, as well as some background knowledge here and here that are really great sort of if you want to dive into the medical world about what's going on for these organelles and how they contribute to disease. But like last time, you want to mosey on over here to the in-class activity discussion board, pop over here to organelles and disease, and you want to create a thread and post your group's answers to this thread. And that's going to be our activity for the day. Going to be trying to link diseases to their malfunctioning organelles. And so you have about an hour, well, you have about 40 minutes to do this. So I'll break you guys off into, again, it's going to be just be random groups. Randomly assign. I want 4 groups, 5 attendees. Break you off into 5 distinct groups because there's 20 of you here. And you'll be able to work on this as a group, discuss it, and post it to, again, that discussion board, which you can find down here. And once you do that, you are all set with me for the afternoon. As I mentioned, your exam will go live at 4, so you can work on your exam after you leave Blackboard. Again, at 4. But again, this is just something to get you thinking about the organelle, about how they contribute to your body as a whole. Because we can learn about these organelles, and that's great. But we want to know how these organelles actually influence you as a system. And I know, like I said, I know many of you are interested in the medical field in one shape or form. So I thought this would be a fun exercise for all of you to do. Now, if you don't understand something in the exercise, like it talks about something, let me know. It might mention something that we haven't talked about thus far. Let me know if you need me to explain it, or you can Google the answer if you so chose. So, does anybody have any questions? And the other thing is, when you post to this, make sure you tell me who's in your group so I know, who did the exercise. All right, so it's going to break you guys off, and I'll come around and check on you. Make sure you talk with one another. Actually interact. This is you might not like that, but it's a really important part of learning, interacting with your peers and stuff like that. So please do interact, talk with one another. You can argue with one another if you so choose, but please do interact with one another. And yeah, all right, group time.